Hello, I'm Bob Fremeau, the founder and acting CEO and CSO of BrainStorm Therapeutics. And we're developing a revolutionary CNS drug discovery platform that is actually NSF-funded and an NVIDIA-award-winning AI platform using human brain organoids to discover new medicines for treating brain diseases. As you know, we're facing a global brain disease crisis. Over 3 billion people worldwide are suffering from a brain disorder. In the US alone, brain diseases are costing $800 billion in healthcare-related expenses. And the real challenge has been the barriers to developing cures has been the high clinical trial failure rates that are driven by inadequate modeling of complex disease biology. There are no reliable animal models that can accurately predict human efficacy in neurodegenerative disorders. In addition to that, Parkinson's disease and Alzheimer's disease, they're not really diseases, but they're syndromes with multiple underlying genetic, environmental, and lifestyle factors contributing to the disease biology. And so we need to be able to develop a personalized medicine approach of developing in vitro human disease models to help identify medicines across these important disorders. And our solution at BrainStorm Therapeutics is to develop a human brain organoid platform. What we're developing, a clinical trial in a dish. So we use patient-derived brain organoids and we screen them with clinically translatable biomarker endpoints so we can identify human efficacy at the very beginning of a drug discovery program. We're also applying biology-first application of AI to develop foundation models that are going to help us identify new targets and underlying biomarkers for assessing clinical trial work. And so, as I mentioned, these patient-derived brain organoids are really transforming CNS drug discovery. They provide a personalized approach of making individual patient-derived organoids that contain native biochemical pathways and highly synchronized spontaneous network activity. And they provide basically a bedrock for doing clinical translational screening. So again, we can identify human efficacy at the beginning of a drug discovery process, and we can de-risk clinical translation using these biomarker-based screening endpoints. And the use of the human brain organoid platform allows us to minimize the unpredictable, unreliable animal models. And finally, the recent passing of the FDA Modernization Act 2 indicates that these human disease models can be used directly to identify compounds for advancing into the clinic, bypassing the need for additional animal experimentation. And so this is really the NSF-funded NVIDIA platform that we're developing at BrainStorm Therapeutics using personalized brain organoids derived from patient-derived iPSCs that recapitulate the complex disease pathophysiology. We do biomarker-based screening using clinically relevant endpoints, and we can identify human biology from the very beginning of target identification through to developing clinical candidates. And finally, by applying an advanced network medicine and advanced computational biology, we can map these disease gene networks to identify novel druggable targets and biomarkers to enhance and de-risk clinical translation. And so this shows that basically we're developing this clinically relevant functional data at scale. So we can grow individual organoids in 3 to 4 well plates and screen them individually with compounds to discover molecules that can reverse the disease pathophysiology. We're using calcium transients as an indirect measurement for network activity shown in the middle panel. And these calcium peaks can be measured by Flipper, showing nice uniform Flipper waveforms representing synaptic network activity. And finally, by doing a subparametric analysis of the shape, frequency, and size of the Flipper waveforms, we can identify heat maps that describe all the different ways in which a disease waveform is different from a healthy waveform. And it's reversing the disease waveforms back to the healthy waveform, which is our drug screening strategy. And so this slide shows that a typical healthy Flipper waveform looks like this. If you make a single point mutation in the gene called MECP2, which is the underlying genetic cause of Rett syndrome, you see an organoid Flipper waveform, which looks dramatically different from that of the healthy organoid. It's asymmetric pattern that shows very different spacing, frequency, and timing. On the other hand, a single point mutation in the same organoid prep in a gene called CDKL5 causes a waveform pattern shown here, which is a very dramatic hyperactivity and a reduction in waveform frequencies. And so we're able to analyze here in a human disease platform, disease-specific functional alterations, which we can then use to do drug screening. And so we are able to then identify, using non-biased approaches, all the different ways in which a waveform differs between the disease versus the healthy. And we show these radar plots here, which identify how the waveforms differ in terms of counts, morphology, peak height, and frequency. And this is really the basis then for our screening activity. And so this slide shows that when we treat the Rett organoids with compounds for 4 weeks, sort of a subchronic dosing paradigm, we can extract, again, these 52 different features that differ between the Rett versus the healthy waveform. And we can then quantify subparametric for each individual waveform feature. We can identify how the drugs affect that. And then we combine that data together to have a global rescue function. So we can report both individual subparameters that are rescued as well as a global rescue function. And so this slide shows that we created these disease models for Rett syndrome, and they show clinically relevant biomarker-based screening. We identified compounds that are able to fully rescue the Rett disease phenotype in these Rett patient-derived organoids. And they do so at brain concentrations that are known to be achieved in the human brain from work that was done by Eisai with donepezil Aricept at the 5 and 10 mg doses. In addition, the rescue that we observe with donepezil occurs at a proper pharmacodynamic effect because we can see the ability of this compound to inhibit acetylcholinesterase in the same organoids. So donepezil is able to rescue the Rett disease phenotype at appropriate brain concentrations and with an appropriate pharmacodynamic effect. And based on these findings, donepezil is now advancing directly to a phase 2 clinical trial being funded by the Department of Defense, being run by Dr. Jeff Newell at the Kennedy Vanderbilt Center, based upon the discoveries that we made with donepezil. We're also putting vorinostat also in the study, the HDAC inhibitor. And so at BrainStorm Therapeutics, we believe that generating these novel human disease organoid models has the potential to provide substantial commercial potential. And at BrainStorm, we're focusing on 2 organoid models: the midbrain organoid model, which contains all the dopamine neurons in the brain, including the A9 neurons relevant to the generation of Parkinson's disease, and the A10 neurons that give rise to the mesocortical mesolimbic pathway relevant to addiction and neuropsychiatric disorders such as schizophrenia. In addition, we have our cortical brain organoids, which we're using now to identify therapeutic candidates for Rett syndrome, CDKL5, and other diseases like autism. So developing these models and validating them, reproducing them provides a substantial commercial potential moving forward, which is exciting for us. And so we're now going to be addressing our current focus really is on addressing the absence of disease-modifying therapies for Parkinson's disease. And it really is the discovery of genetic forms of Parkinson's disease, which provides a path forward to disease-modifying therapeutics. And so mutations in the GBA1 gene, which codes for lysosomal enzyme glucocerebrosidase, or LRRK2, which is an enzyme that plays a role in mitochondrial function, these are risk factors that cause Parkinson's disease. So we made brain organoids from both GBA1 and LRRK2 organoids, and we're able to demonstrate a robust and reproducible loss of dopamine neurons in these organoids. This work was done in collaboration with Jens Schomborn and his team at Organotherapeutics. And so in different patients and different differentiation batches in Luxembourg and in San Diego, we were able to identify a dramatic loss in dopamine by staining for tyrosine hydroxylase, the rate-limiting enzyme making dopamine, and a specific marker for dopamine neurons, but also by measuring dopamine secretion into the organoid media. So we see a robust and reproducible, stable platform that we can measure dopamine neuron loss in these organoids, providing a basis now for screening for medicines that can reverse dopamine neuron degeneration in these organoids as a therapeutic for disease modification for Parkinson's disease. It's really important. One of the outstanding features of our platform is our ability to generate stable, reproducible organoids. One of the drawbacks in the field has been the variability in organoids. We're actually working on spheroids. And so we have established now by doing both bulk RNA-seq and single-celled RNA-seq across many different patients and many different batches, we see a high conservation of gene expression in these organoids. So in addition to having a stable, reproducible, functional loss of dopamine neurons, they also show a dramatic conservation of gene expression, both at the single-cell level for cell composition and the bulk RNA-seq level showing overall gene expression patterns. The correlation coefficient for the bulk RNA-seq data is 0.92, showing a striking conservation. In fact, our principal variant component analysis demonstrated that the batch variations account for less than 10% of variation across patients in differentiation batches. And so having this really high quality both bulk and single-cell RNA-seq data from individual LRRK2 and GBA1 patients provides an opportunity now to use this data to do more detailed analysis of computational biology using AI machine learning approaches. And it really is the quality of the single-cell and bulk RNA-seq data that allows us to use AI approaches to develop foundation models. And so what our team has done, Jun Yin in the audience and Maya, we formed a team that took part in a Lux Capital hackathon. And over the course of a couple of weeks, with a very talented team, we're able to fine-tune sort of pre-existing foundation models. We fine-tune those models with the RNA-seq data that we identified from our LRRK2 and GBA1 organoids, and we developed a gene expression network pattern, which allows us to then identify genes that are upregulated and downregulated in both LRRK2 and GBA1 organoids, and they ended up falling into identical clusters. So they're clusters of differentiated genes in these organoids. And it turns out through the analysis that Jun and Maya did, these clusters of altered genes correspond to specific known biological functions in the cell. And it's the ability to map the gene expression patterns to known biological functions, which really help us identify that these organoids are able to predict the known dopamine neuron loss and the gene expression loss in actual patients. And that's, I think, probably the most profound observation we have made so far. And so you can see here that the GBA1 organoids show clustering of genes relevant in cell differentiation and in neurogenesis. What in the world are genes in cell differentiation and neurogenesis doing in a 60-year-old human PD brain? But we believe that our work has actually changed the paradigm now, that the seeds of destruction in a dopamine neuron are planted already shortly after or during birth. And so in addition to that, we're able to identify organoid-specific effects. So the GBA1 enzyme, which functions in lysosomes and metabolizes lipids, we see, again, a cluster of genes involved in lipid metabolism. LRRK2, which is known to regulate mitochondrial function, we see clusters of genes that are involved in oxidative phosphorylation. And the really exciting outcome of our work is that the cell differentiation and neurogenesis pathways that we see both in GBA1 and LRRK2 organoids were also observed in human dopamine neuron data sets, differentiation and neurogenesis. And so our organoid-based foundation model is able to predict the generation of dopamine neurons that are observed in the human brain. So with that, I'd like to take a step back and talk about the company. We've actually been able to generate some we were founded in March of 2023, a little over a year and a half ago now. In that time, we've had our platform validated by the Department of Defense funding of our clinical trial that's being done in Rett syndrome, the donepezil and vorinostat work. We've actually, as I mentioned, had an NSF-funded phase 1 award that supports the development of our Parkinson's disease organoid drug discovery platform. And we just won the NVIDIA award for the best platform, best AI platform as part of the Lux Capital hackathon. And so our team really is a very strong leadership team with deep expertise, both on the executive founding team, but also our scientific advisory board in CNS drug discovery, stem cells and brain organoid, and data science. We've received letters of interest from KOLs and drug developers that want to use our platform to help them pick their development candidates from their own leads. And we have developed strong links and relationships with patient foundations, including IRSF and the Lulu Foundation. And so we're currently in the process of applying to incubators and accelerators, and we're trying to raise seed funding as well as developing strategic advance our portfolio. And so as I mentioned, I'm very lucky to be joined by both Jun and Maya as the co-founders for BrainStorm Therapeutics. So I spent 10 years as a scientific director at Amgen. And during my time at Amgen, my colleagues and I advanced 12 compounds into the clinic, and they all failed. They all failed primarily due to lack of efficacy. And that was my first indication that the way we're doing CNS drug discovery is broken. Using animal models is a very poor way of identifying medicines for humans. And so we think by having these human disease models, we have the chance now to really make a difference. Jun is also a colleague of mine. We both worked at Amgen. We didn't overlap, but Jun comes with a very deep expertise in applying machine learning and AI approaches to doing drug discovery, and he helped to contribute to building the genome analysis unit at Amgen, the first organization like its kind in any pharmaceutical company. And Jun's work helped identify both target discovery, target advancement, and his work led to the approval of medicines across our Amgen portfolio. And Maya is the most recent member of our team and co-founder. She's a recent Ph.D. graduate from Jean Yo's lab, one of the really pioneering labs at UCSD. Maya brings both an important organoid expertise, developing brain organoids, but also she has deep expertise in both machine learning and AI approaches for doing drug discovery. So she really nicely complements myself and June as well. And again, as I mentioned, we have a very outstanding scientific advisory board. Alison Moutry is one of the pioneers in developing iPSC-derived brain organoids. James Traynor, Mark Norman, and Dotto all worked together with me at Amgen. James was the key person that was involved in driving most of the clinical trials. He's a very excellent CNS drug discoverer. He's the guy at Amgen who led to the marketing of Aimovig, of the migraine compound for Amgen. Mark Norman is a very talented medicinal chemist who's advanced more than a dozen compounds into the clinic and has many, many publications and patents. Dotto, again, is a really expert computer scientist, and he's an expert at applying sort of software engineering, machine learning, and AI approaches to drug discovery. And Blake Anson is a colleague of mine from our former role at Stemonics and Viant Bio. Blake is now working at Fujifilm Cellular Dynamics, and he's an expert at developing stem cell platforms for doing drug discovery. And so our hope now is that we can raise $6 million in seed funding over the next 12 to 18 months. Our goal now is to complete screening of our GBA1 organoids to identify hits that are able to rescue dopamine neurodegeneration as a starting point for doing lead optimization. We also have identified an epigenetic target that's able to rescue the Rett disease phenotype. It's an epigenetic writer. And it turns out molecules that are known to interact with that target have shown efficacy in 5X FAD Alzheimer's models. So we think we can use clinical trial work in Rett syndrome to identify compounds that can then expand into neurodegenerative disorders. And we also have identified both hits and repurposing medicines for CDKL5 deficiency disorder, otherwise called CDD. CDD is the most common rare genetic, it's hard to say this, developmental epileptic encephalopathy. And so compounds that work against CDD are likely to also have efficacy across the other DEEs. And so with that, I'll stop, and if I have time left, take any questions.
We'll let you have a question. Who's got a question about brain organoids and BrainStorm Therapeutics and anything?
Hi, I'm not one of the judges, but I had a question anyway. I noticed that in your pie chart of the source of the variance between organoids, the largest variance actually came from donor to donor variability, right?
No, actually not.
Maybe I'm misremembering that. Yeah, donor is 41.2%.
Oh,
right?
Donor meaning GBA1 versus wild type.
Oh, I thought that was disease status. Okay,
No.
Sorry, it wasn't clear.
Oh, it's okay. It's shown here. So these are the controls, and these are the GBA1. And so we have multiple patients and multiple batches per patient that constructed the heat chart. And so the major variation is from donor, which refers to the disease gene status.
So what's disease status?
Pardon?
What is the orange disease status?
Different patients, donors or control donors. That actually means that our system is sensitive enough to differentiate different donors in the experiment. And if you look at the heat map carefully, you'll see the stripes actually on the heat map. And that means like the data actually from the same donor. And remember that earlier we showed that if the patient are from carrying a GBA1 mutation and NERC2 mutation, our system is sensitive enough to identify the specific signaling pathways pointed to that particular genotype.
Is that helpful, Reed? Thank you, June.
All right.